Okay. Oh, excuse me. That was a big yawn. I apologize. Okay, so back to recycling the components of red blood cells. Let me just be sure that I'm still sharing my screen, which I don't think I am. Okay. So now we have this iron. And we get iron from our diet, obviously, but we don't want to get rid of this iron. We would like to use it for production of more red blood cells. And if we have extra iron, we would store it in case we need it in times where maybe we're not getting the proper amount of iron in our diet. So when we eat meat or food that contains iron, it gets absorbed into our bloodstream, and you'll see here, right here, is a carrier protein called transferrin. So the iron that we ate will be carried on transferrin to the bone marrow or to be stored in the liver. After we have our aged red blood cells that are broken down in the spleen and the iron is released, that will also be carried on transferrin and can be stored in the liver as ferritin, or can be taken back to the bone marrow to be incorporated into red blood cells. So transferrin is our carrier protein of iron in the bloodstream. Ferritin is our storage protein. So when the iron binds to apoferritin, which is the storage protein, it becomes ferritin. So that's our storage in the liver. And then we can reuse iron after the red blood cell is destroyed in the spleen in order to produce more red blood cells. And we try to keep a constant iron hemostasis. So now we have to look at the types of anemias and some of these things that we've talked about, like what happens with red blood cells when they're recycled and what is the life cycle after we try to produce a red blood cell will come into play, because that will help us figure out what is the cause of an anemia. So we can look obviously at the factors that the patient, their signs and symptoms of other, problems that are commonly associated together with types of anemias, but we can also look at their blood tests for clues as to what kind of anemia is taking place. So we talked about the white blood cell and the differential. Now we're going to talk about the red blood cell count, hemoglobin and hematocrit count, and the red blood cell indices that help us distinguish types of red blood cell disorders. So first, we look at the red blood cell count, and it would either be normal. If there's too much red blood cells, that's considered polycythemia, or if there's decreased red blood cells, that's anemia. So there are other tests we can look at that will give us more information about the red blood cell itself. So we said that the red blood cell has hundreds of hemoglobin molecules. So we can measure the hemoglobin content in the blood, and that gives us or lets us know a couple of things. One is we would expect that if there were less red blood cells, then obviously there would be less circulating hemoglobin, because hemoglobin is in red blood cells. It's also possible that we could have a greater loss of hemoglobin than would be expected, meaning that my red blood cell level is low, and so I'm expecting my hemoglobin level to be low. But if my hemoglobin level is even lower than expected, that means that of the red blood cells that are present, they don't have the same amount of hemoglobin in them. So the quality of the red blood cell is also bad. Hematocrit is telling us the volume of cells, the volume of red blood cells in 100 cc's of blood. So when we talk about this in this particular unit, I want to change the way you have thought about it probably as a nurse. So here's my vial of blood. If I spun it down into its component parts, like that first picture we saw where we saw the plasma, that white stripe of what would be white blood cells and such, and then the red blood cells. This percentage of red blood cell in a volume of plasma is hematocrit. So if the person has anemia, then they have a decreased number of red blood cells, then I would expect that the percentage of red blood cells would be lower because there's just less red blood cells in general. Now, sometimes, when you are working and the person that's inpatient in the hospital has been there a while and they have reasons to have decreased production of red blood cells, or because maybe they've suffered a loss and we're trying to get them to pick up, but perhaps their fluid management has not been done very well and they are fluid overloaded. So their hematocrit can seem low due to dilution. But in this case, we're not talking about it in terms of fluid status. We're talking about it being low because the actual number of red blood cells is low. Just keep that in mind for like an exam or case studies. There are obviously other reasons why the hematocrit can be low based on fluid status, but in this situation, we're talking about it in terms of anemias themselves. Reticulocytes are those immature cells that the first time that the red blood cell is extruded from the bone marrow into the circulation, it has to go through another 24 hours of maturation to become the erythrocyte. The reticulocyte is letting us know something about the speed of production. If there's an increase in number of immature red blood cells, then we're obviously trying to make up for some sort of loss. So erythropoietin must have been released to increase production of red blood cells. And so if we're looking at the percentage of mature erythrocytes to reticulocytes, if the reticulocytes are higher, then the production rate has increased. The other thing that we can look at when looking at red blood cells is what are called red blood cell indices. And these are looking at the quality of the red blood cell itself. Mean corpuscular volume is talking about the size of the red blood cell, and mean cell hemoglobin, or mean corpuscular hemoglobin concentration, is telling you, so this is size, this is telling you how much hemoglobin is in the actual red blood cell. So these are sort of a quality indicator of the red blood cell. So we have these red blood cells, but are they adequate? The ones that we have, is it just that we don't have a lot? Are they also like subpar? So we can look at all those things together with the patient's story to try to classify an anemia. So anemia in general is a reduction in the total number of erythrocytes circulating in the blood, or in the quality or quantity of hemoglobin. So either we have a decreased red blood cells, and we have a decreased red blood cell and hemoglobin. And it's either impaired production, so that any of the things that we needed to make the red blood cells, like proteins, vitamin C, all the B vitamins, copper, iron, there could be something missing that we need for production. We could be losing blood, either acutely or chronically. We could have an increased destruction of erythrocytes. Maybe they're not lasting as long as they're supposed to, or it could be a combination of those things. So we will see that the patient has, or the person has, a decreased red blood cell count, and we will also look at both the size and hemoglobin content of that red blood cell. If the red blood cell is large, it's called macrocytic. If it's small, it's microcytic. Sometimes that's related to the reticulocyte count, because reticulocytes are smaller. But sometimes it has to do with the lack of component parts that we need to make the red blood cell. Hemoglobin content is going to tell us the color. More hemoglobin, the darker the red blood cell. So normal amount would be normochromic. Hypochromic means that the red blood cell is lighter, and that is because there's less hemoglobin in each red blood cell. So the first anemias we're going to talk about are related to deficient production. So we're not making the correct number of red blood cells. Excuse me. And typically the main reason for that is iron deficiency or deficiency in the B vitamins. So iron deficiency anemia is by far probably one of the most common anemias. And there's two reasons that this can occur. One is that you're not getting enough iron in your diet. It is a little harder to be iron deficient because we have a lot of iron fortified grains. So although getting adequate meat in the diet that's more expensive, and so you could have nutritional deficits. It's possible that you could get iron from other products. So malnutrition is possible, or it's because of chronic blood loss. So if we're dependent on the fact that we're recycling iron and that we have a storage capacity for iron, if the red blood cell is lost prior to it aging out by the spleen and letting the iron be recycled, then as that happens over time, we will lose iron stores and end up with an iron deficiency anemia. So one of the reasons that this can happen, one of the big reasons, is a slow GI bleed, either from a chronically oozing upper GI ulcer or cancer or oozing blood loss in the small or large bowel, or from very heavy menstrual periods. So in the beginning of this chronic blood loss, you will start to notice a decrease in red blood cells and a decrease in hemoglobin and hematocrit just because there's overall decrease in the number of red blood cells. But when you look at the red blood cell indices, because at the time that we started bleeding, we still had iron storage in the liver, the cells that are produced are still normal sized and normal color. So in the beginning, a chronic blood loss will show an anemia that is normocytic, meaning the mean corpuscular volume is normal, and normochromic, meaning the mean corpuscular hemoglobin concentration is normal. Later, as we lose iron storage and then there's going to be a change in the synthesis of red blood cells because of the loss of the iron. So it's harder to make red blood cells, and the red blood cells that we have are not going to be of the same quality. So first, there's going to be a change in the color where there'll be low red blood cell, even lower hemoglobin because we're losing that. And we're going to start to see at first a normocytic hypochromic anemia, and eventually it'll even, if this happens over time, microcytic hypochromic anemia. So then the question becomes, if you see this patient and they have developed an anemia and the red blood cell indices suggest that the cells are getting smaller and lighter, you could confirm your suspicion by doing iron studies. And these are this is what is called an iron panel. So you would be looking at the serum iron level, the serum ferritin level, which is apoferritin bound to iron, which becomes ferritin. There should be a small percentage in the bloodstream. Most of it is, of course, in the liver. There should be a small percentage of free iron in the bloodstream. Most of it should be places where it needs to be used. And then transferrin and total iron binding capacity are similar. So we know that transferrin is our carrier molecule. It's our carrier protein that carries the iron that we eat in our diet to the liver or bone marrow, or the iron that's broken down in the spleen to the liver or bone marrow. If we begin to have a decrease in iron or red blood cells, the automatic response of the liver is to increase production and release of transferrin. This protein is made in the liver, and the thought is, well, maybe the reason we're not getting the iron we need is we don't have enough carrier protein, so I'm going to make more carrier proteins. So people with iron deficiency anemia tend to have low iron, low ferritin, and an increase in transferrin, which also means an increase in total iron binding capacity. So I like to think of transferrin as, and as you get older, some of your references sort of age out as well. So I'm not sure how many people remember playing the game, the board game, the Game of Life, which has those cars that have like the sights for pegs. So I don't know if people remember that anymore. But anyway, this carrier protein has open sites for iron. If iron isn't present, then there's more open slots. There's more seats available in the car. The more transferrin that's present, the more capacity to bind iron is present. And so over time, as I lose my iron stores, my total iron binding capacity will be elevated because there's room for iron. Thank you! There's my classes where everyone looks at me like I'm crazy. I totally love that game. So, if a person has iron deficiency anemia, in the beginning, there might not be a change in any of those numbers until the storage of iron is lost. And then we'll have a decreased iron, decreased ferritin, which is storage, an increase in carrier proteins, which is transferrin, and then increased space for binding of iron, which is an increased total iron binding capacity. So that's one type of anemia. Another type of anemia can develop. It's sometimes called megaloblastic anemia or macrocytic anemia, which means we have a decrease in blood cell count, and it's related to impaired production of red blood cells, and that's not related to iron deficiency, but related to a deficiency in the B vitamins that are necessary for DNA synthesis. That's part of the cellular differentiation when we're going from the erythroid progenitor cell to the mature erythrocyte. So that picture way up in front here, where we had this committed progenitor cell, here the nucleus, this is the nucleus extruded. Here the nucleus is smaller. This step, the nucleus is really big. We need production, we need the B12 and folate entered in this part of the red blood cell production. If not, then we miss a step and we end up with bigger red blood cells. So we're going to produce less blood cells because we don't have the parts we need. And the red blood cells we make are going to be larger. So B12, so how do we get B12 and folate that we might not have enough of that in order to make red blood cells? Either we have to absorb B12 in the stomach. Some of it is absorbed in the last part of the small intestine in the ileum, but most of it is absorbed in the stomach. And folate is absorbed in the first part of the small intestine, the duodenum or duodenum, whichever way you like to say it. So it could be that we have a dietary decrease, food insecurity, or potentially these B vitamins tend to be low in association with alcohol intake. Or Alternatively, if it's not a dietary issue, there could actually be a gastric or intestinal problem that's changing the absorption of these particular nutrients. So in terms of B12, B12 deficiency is the most common cause of macrocytic or megaloblastic anemia. And B12 deficiency is caused because there is a decreased ability to absorb B12 in the stomach. So it could just be that the stomach itself is smaller because maybe we've had surgery, so we've lost surface area, or the stomach has changed related to medications we've taken or infections we've had. But the other type of B12 deficiency is a special type of anemia called pernicious anemia, in which either congenitally or later on through an autoimmune disorder, we have a lack of intrinsic factor. So when we eat foods with B12, intrinsic factor is needed for the stomach cell to be able to absorb, in order for absorption of that B12 in the body. So gastric parietal cells are what make intrinsic factor, and intrinsic factor binds to B12 in the stomach and then is absorbed in the last part of the small intestine. So if our B12 that we eat never encounters intrinsic factor in the stomach because our parietal cells aren't making it or it's being destroyed, then as that B12 works its way down the small intestine, it won't be absorbed. It needs the intrinsic factor in order to do that. So I could have an overall lack of intrinsic factor, and that's the reason why I have B12 anemia. And so in that case, I obviously couldn't give somebody an oral supplement of B12 because it doesn't matter how I give it, whether it be a pill or food. If I don't have the intrinsic factor from the parietal cells, then I'm not going to be able to absorb B12 later on in the small intestine. So then that person has to get it directly deposited into the bloodstream. That would mean like IV or IM supplementation. The other type of macrocytic or megaloblastic anemia is a folate deficiency anemia. So that's the other B vitamin. And this is also essential, again, in that step where we're changing the quality and differentiation of that DNA material in the nucleus during those steps of maturation of the red blood cell. So we get folate from the duodenum. It does not need a facilitating factor, so it's not some kind of congenital or autoimmune disease where we're lacking that facilitating factor to absorb the folate. It has to do with either we're not getting enough. It's usually related to chronic malnourishment and alcoholism. We know that folate in general is very important for nerve function and neural tube development, and so it's often recommended to people who are pregnant or wish to become pregnant in order to protect against those kinds of disorders. So besides the symptoms of anemia, the person would have a low red blood cell count, a low hemoglobin and hematocrit. And then if I looked at their indices, there's enough iron, so the blood should be normal chromic. But because this is a macrocytic anemia, I would expect that the red blood cell will be larger. MTHFR mutation it's a alteration in how folate is used, because folate is usually the treatment, folate supplementation, but MTHFR mutation changes clotting. So this, you can treat it with folate and Coumadin, depending on how severe the mutation is. But the issue that the patient complains of with MTHFR mutation is excessive clotting. So it's partially there, but not related to anemia. Don't want to type in something else. There's another term for that mutation if you just look at those letters. I can't say it because Alverno is a Catholic school. I think that's kind of an oversimplification of it because you can treat it with folate, so the body has to be able to do it. I myself do not fully understand what the mutation of the MTHFR gene does and how its use of folate occurs. It's a change in the use of folate in the production of clotting factors, though, not red blood cells. I would have to talk to a genetic counselor. Yes, that is true. It's just not, it just doesn't, those people don't have anemias. They don't have megaloblastic anemias. It has to do with clotting. Okay, anemia. So besides the fact that they have the red blood cell issues, they're also going to have issues potentially with nerve structures because of the relationship between folate and nerves itself and myelin of the nerves. So sometimes they have feelings of paresthesias or numbness and tingling. They can have some paralysis. Ataxia means that their movement, they are walking off balance. They can have confusion, sores or changes on the sides of their mouth or inside their mouth, and sometimes paresthesias and pain inside their mouth. So either of those anemias, B12 or folate anemias, are both macrocytic anemias. So I would expect that there's enough hemoglobin because iron isn't the issue, but instead they're macrocytic, they're larger cells. However, you can certainly have both at the same time because, you can have more than one thing wrong at the same time. The other types of anemias are types of anemias that increase hemolysis. So that means our red blood cells are not making it through the spleen and are being broken down early. And usually those are extravascular hemolysis issues. It's a problem with the production of the red blood cells that's usually related to genetic mutations on the parts of the gene that make hemoglobin. You can get intravascular hemolysis as a result of other toxins, like infection. Toxins sometimes cause hemolytic anemias, an injury or transfusion reaction. We're going to talk about the types of extravascular hemolysis that occurs related to genetic mutations. So sickle cell anemia is one such anemia. This is an inherited recessive anemia. So that means both parents have to at least be carriers of this disease in order to give one of their offspring this disease. When you inherit from your parents, one side is from your mom and one side is from your dad, you would have to get sickle cell from both sides in order to actually have the disease. If you just inherited on one side, then you're a carrier. You're not going to display the disease. This, interestingly enough, the reason why sickle cell anemia is found in certain ethnicities is because there was an evolutionary reason to develop sickled cells. In places like in Africa or near the equator, Mediterranean area, where mosquito diseases like malaria are endemic, to have sickle cells is protective against parasitic infections of the red blood cells, which is what malaria is. So this type of trait, this inheritance, was developed because people who had sickle cell were more likely to survive or not have malaria infections. However, it doesn't do well in other climates where malaria isn't endemic and when it's really, really cold. So there is an abnormal structure of hemoglobin because there's a mutation in the beta chain of the hemoglobin molecule, and this substitution of one of the amino acids causes the chains to be sticky. So here's my, oops, I forgot I have to keep drawing. Here's my hemoglobin molecule. It's alpha, alpha, beta, beta chains, and I have my heme units, and oxygen is on all these spaces, and when it diffuses off to help oxygenate the tissues, carbon dioxide should come on here, or at least it won't change the actual structure of the hemoglobin. When somebody has sickle cell anemia, and we have our chains, and here's our beta chains, the one that has a mutation, if the oxygen comes off, instead of allowing to have that open site, these two will stick to each other. And so instead of keeping a normally biconcave disc of a red blood cell, we end up with a sickled cell because these hemoglobin molecules are sticking to each other, and that happens after oxygenation. So here's our point mutation where we change one amino acid, and that's enough that when this the hemoglobin on this red blood cell deoxygenates or releases its oxygen, it can get that sickled shape. Occasionally, that is reversible, so the next time it picks up oxygen, it'll go back to the shape it's supposed to be. Sometimes these cells will be irreversibly sickled, so they're sticking to each other, and they can be destroyed early because when they make their way through the spleen, they're gonna get stuck in there. So they could get sequestered in the spleen, causing splenic enlargement, and they're going to be broken down more in the spleen. Or these cells do not make their way through the circulation as well, especially the microcirculation, which can lead to occlusion and tissue ischemia infarction, that coagulation necrosis in certain areas. So having sickle cell anemia can be extremely painful and can greatly reduce a person's life expectancy. People with sickle cell anemia, life expectancy is down into the 50s and 60s instead of being in the 70s. They can get strokes and blindness. Any area can have infarction. Their spleen can be enlarged or infarcted, and that's why it can get smaller over time. They can get painful avascular necrosis where the head of the femur dies, different skin ulcers. You'll notice here that it says iron overload. So people with sickle cell anemia, so they have an anemia, so they're going to have a decrease in red blood cells, a decrease in hemoglobin and hematocrit, just because of the overall loss of red blood cells. They may have an increase in iron, and they'll likely have an increase in bilirubin. Why? It's because of the higher rate of red blood cell destruction. So if I'm destroying red blood cells in the spleen, then I'm releasing the components of the red blood cell into the bloodstream, which normally I would use for recycling. So I'm going to have a higher amount of iron in the bloodstream because of that. I'm going to have a higher amount of bilirubin. Those are the components of the heme unit. I'm going to lose the iron, and I'm going to lose the porphyrin that gets changed through enzyme reactions into bilirubin. And my liver has the capacity to conjugate that bilirubin to allow for removal. But if there's too much bilirubin, then I'm going to overwhelm the capacity of my liver. So that is one thing that I will notice with the hemolytic anemia. Another type of hemolytic anemia is a group of inherited disorders called thalassemias. These are also common um, in other, more common in Mediterranean and Persian ethnicities. Um, they are, they could be a problem related to the alpha or beta chain, and they're classified by where the mutation is located, whether it's a beta or alpha thalassemia. So is the mutation on the beta globulins or the alpha globulins? And is it major or minor? So are both beta chains mutated or are both alpha chains mutated? Because that means that the whole hemoglobin molecule is, one side of the hemoglobin molecule isn't going to work effectively. So major beta thalassemia means both beta chains are affected. Minor beta thalassemia means just half of the beta chains are affected. But either way, we end up with an increased destruction in the spleen. So we still have anemia, and we have an increased chance of iron and bilirubin in the bloodstream. But this particular disorder does not cause the sticking of the beta chains. It does not cause the sickling. So you don't have the ischemia associated with that we get with sickle cell. And the last thing we're going to be able to talk about before we run out of time here is polycythemia, which is an absolute increase in production of red blood cells. So it's either a primary disorder of the bone marrow in which there's increased proliferation of red blood cells, like as in a cancerous growth, or secondary to hypoxia, because when we have a decrease in oxygen saturation, that sends a message to the kidney to release erythropoietin, which will increase production of red blood cells. So a primary polycythemia means there's some kind of tumor. There's no regulation of growth, and that primary disorder is called polycythemia vera. But secondary is related to an increase in erythropoietin response to hypoxia. This is more common. It could be an erythropoietin secreting tumor, but usually you might see it in cases where people would normally be oxygen deprived, such as people who smoke. They are oxygen deprived, and so they will increase production of red blood cells. People that live at altitude have a decreased oxygen concentration in the air. It would be normal for them to have an increase in red blood cells. The spleen may become enlarged because there is more red blood cells, so there might be sequestration of red blood cells in the spleen. You're increasing the amount of cells in the blood, which increases your risk of clots. So the last portion of this is the blood coagulation, the process of hemostasis and problems with coagulation. So I will have to just post that as a recorded lecture, which I've had to do before, so I just have to pull that up and download it onto our Moodle site. I have recorded this, so I will also download that onto our Moodle site. So it's 9:30. Is there any questions about anything we've talked about so far before we end it for tonight? Obviously a lot of correcting to do so we can keep on track of stuff.